Hi, everyone. Sorry for a few minutes of delay. Thank you very much for joining. My name is Kiana Aran, and I'm the CSO and co-founder of Cardea Bio. I will be moderating this session. And along with Dr. Qi, Dr. Gootenberg, and Dr. Abudayyeh, we will be presenting a diverse application of CRISPR in this session for you guys. In today's talk, I'm going to talk about CRISPR-based transistors for specifically for CRISPR quality control applications. And I'm going to start with the vision of Cardea Bio and what is it that Cardea does. The vision of Cardea Bio is really looking into biology as system biology. Biology is systems of systems, which are communicating with each other in a dynamic fashion. For many years in life sciences, we've been taking snapshots of these dynamic events at one time point. And we've been using optics to understand biology and define different type of pathology from these snapshots information. But to truly understand biology, we need to take a system biology approach. We need to understand and get an insight to multiple different analyzed and multi-perspective biological data. And we need to do that close to real time so we can understand these dynamic events that is happening between these biological systems. And then we can have a flow of information that can, and not only a snapshot data set. This will allow us to understand biology in context. So, and this will be very important for fundamental discoveries in biology. So how do we do that? In order to do that, and achieving this vision really requires that we can measure multiple different type of analytes at the same time and get all of these large volume of information and transfer it in a seamless way to computers so we can do machine learning analysis on those and connect those to wireless networking and develop personalized devices. To achieve this vision, this is not doable with optics because optics has a lot of limitation. Like, the wavelength of optics is very limited, so you cannot detect things that are smaller on a certain size. And this is only achievable by developing highly scalable, cost-effective electronic system where we can seamlessly transfer this huge volume of data to computers so we can perform machine learning algorithms on them and analyze the data. And this is what Cardea does. Cardea basically provides an infrastructure where we combine tech, we provide a tech plus bio infrastructure where we can directly streamline multiple different information from basically multi-omics data to computers, which allow us to have access to new biological information. So to do that, we use graphene field effect transistors. So graphene field effect transistors are transistors that have a conductive graphene material between two of their terminals, namely drain and source. And this conductive material it's functionalized with the biological molecules to interact with the biology of our interest. And basically, the way that we biologically activate our graphene channel will allow it to interact with our biology of interest, whereas it's a protein, RNA, or DNA, or any type of biological molecule. This interaction can change the conductivity of the graphene material, which we can sense it in a real-time manner using our graphene transistors. Graphene transistors are also superior to silicon-based transistors because they have high electron mobility. That means that the signals can be measured much faster, and they also don't interact negatively with biology. That you can directly connect them with biology. Whereas with silicon-based transistor, you really have to have a layer of oxide layer or other type of layers to protect the silicon material itself, and the biology also it's impacted by the effects that the silicon could have on it. But then the biocompatibility of the graphene does not have these problems basically. So the way that we make our transistors specific to a certain biology, we use a capture molecules on the surface of our graphene material. This capture molecule can be an enzyme, protein, antibody, a CRISPR enzyme, basically anything that you can imagine. And the interaction of this biology with our target analyte will generate that signal. The selectivity of our sensors comes from this capture molecule. And the sensitivity of our transistor comes from the biology itself. How good is the biology? If it's an antibody, how good is that antibody? If it's an enzyme, how well that enzyme works? And of course, we can optimize our sensors and sensor design and operation parameter to improve the sensing of our transistors. Our transistor, again, are true multiplexing system, because you can envision having thousands or hundreds or thousands of these transistors on a single chip. And each of these transistors can be functionalized with a specific capture molecule where you can detect RNA, proteins, DNA at the same time in the same system. You can also make them high throughput because these are electronic systems and can be easily reconfigured to different type of form factors. The picture you see on the right is basically shows the evolution of our chips, and it's really following and outpacing the Moore's law. As we keep generating new generation of our chips, the cost goes cheaper, and the fabrication becomes easier and more understandable for us. So the chips that the final chip that you see on the first chip from the right is the chip that we currently use in Cardea. We have enabled a lot of our partners to actually use our technology for a variety of different applications, from antibody testing. We have worked with CDC to detect Zika. We have worked with a lot of industrial partners to detect cytokines. We have done the DNA sensing, and we allow our partners to basically use our system to many different applications, because we want to examine our chips and improve them for multi-analyte applications. One of the most recent technologies that we developed last year, which was published in June 2019, was a technology that utilized CRISPR on the transistors to detect the target sequences. Basically, when we looked at CRISPR, what we saw was not only a technology that could be used for gene editing, but also a technology that could use as a search engine. The CRISPR-Cas complex is capable of finding its target sequence within the whole genome without amplification, without doing any modification with the DNA, before we can do any editing on it. So the first thing that we saw with the CRISPR complex is its power of its searching power, basically. So the way we envision this, can we actually utilize the power of CRISPR on chip and basically just monitor it, just see when CRISPR binds and see when CRISPR cleaves, and not use any sort of amplification or not modify the target DNA, and see if we can capture the function of CRISPR using our transistors. So that led to the first electronic sensor that was CRISPR powered, and we were able to detect DNA sequences without amplification. Basically, it was not a rocket science. We used a technology that was developed by bacteria over millions of years as a technology in our transistors, and we utilized the power of CRISPR to develop our sensor. So the way that the CRISPR chip works, we basically immobilize thousands of CRISPR complexes on the surface of the graphene channel. First, we immobilize the Cas enzyme, then we passivate the rest of the graphene surface, then we programmed the Cas enzyme by adding the guide RNA, and then we introduced the whole genome sample to the DNA. When I talk about whole genome, that means there was no amplification done. So that's the biggest step in sample prep, especially considering that PCR amplification requires an infrastructure. We utilize the technology to detect multiple different deletion in different cell types, which was published last year. We have another manuscript coming up, which we are developing, which we have tested the sensor for monitoring single point mutation in sickle cell disease model. We have compared different type of guide RNAs at different lengths to monitor the binding efficiency using our system. We have compared different Cas variant and high fidelity Cas enzyme in their efficiency using our system. We have detected RNA binding to Cas protein in an automated fashion and have added software package to analyze the data with our system, which I'm going to go through briefly here. So this was what we published in June 2019. Again, that was the first CRISPR-powered electronic sensor reported, where we actually were able to detect two deletion and 2 exon in patients with Duchenne muscular dystrophy. So basically, we developed CRISPR chip that was detecting deletion in exon 3 in dystrophin gene, and we developed a CRISPR chip that was detecting a sequence in exon 51 in dystrophin gene. And we had samples from patients that were lacking either lacking exon 3 or exon 51. So as you see, the plot on the left shows that the CRISPR chip that was targeting exon 3 was able to detect the presence of exon 3 in healthy samples, and was able to detect the deletion of that sequence in exon 3 in disease models where they had exon 3 deleted. And we also detected the presence of these exons in samples D, E, and F, where they were lacking exon 51, but they had exon 3. We had one false negative, which was a true-false negative, basically. The plot on the right shows a CRISPR chip that was targeting a sequence in exon 51. The result shows that the chip was able to detect the presence of that sequence in healthy samples and in disease samples where exon 51 was present, but exon 3 was lacking. And the lack of the sequence in exon 51 was detected in all 3 samples, D, E, and F. To define the threshold, we collected the largest volume of genomic sample that could be acquired by utilizing buccal swab, which is about 2.8 microgram of non-target DNA with our chip, and this is how we defined our threshold. So you can envision by having hundreds of these transistors, you can really sequence and detect different targeted region of the gene and identify their presence or absence. And this is how we envision with CRISPR, because CRISPR is so versatile, and by changing the guide RNA, you can basically target different region of the chip. And by true multiplexing capability of our transistor, you can envision a system that could use the power of CRISPR to monitor long regions within the gene. This is another set of data where we have been able to show the single point mutation in patient sample with sickle cell disease. And single point mutation without amplification is not easy to accomplish. It's a very difficult task. And again, we have improved our sensor in terms of data analysis, but the main power comes from the CRISPR, comes from the improvement in the guide RNA design and on how to best design this guide RNA to discriminate between single point mutation. And we have utilized those advancements that biologists and people who are using the CRISPR for sickle cell disease to select the best guide RNAs and look to see if we can discriminate between healthy genomic and sickle cell disease samples. And again, as you can see, we were able to clearly distinguish between sickle cell disease and healthy samples using our chip. We did a little bit more data analysis for this paper, which I'm not going to go into deep details. We're currently working on zygosity. We are very interested to use our platform to identify the zygosity and see how complex selection, protocol selection in, for example, your buffers, your ions, how do they impact our chip in selection, in being able to discriminate between homozygous and heterozygous type of samples. And we are very excited to see some of our results, but this is still work in progress. One of the major things with our technology, because we are real time, we can monitor the whole process of functionalization from when we put the Cas enzyme through when we introduce the guide RNA. And this has led to our first product, which we call CRISPR-Bind. As you know, many of people are monitoring, are modifying their guide RNA, or they're using different type of Cas enzyme for different type of applications. And it's important to make sure that those modifications are not impacting the complex itself, are not impacting the stability of the complex. Our first product, which was launched a few weeks before, basically it monitors the guide RNA binding to the Cas enzyme, which can be utilized by researchers to make sure that their modification is not impacting the design. Our next generation, which are CRISPR amps, which will be monitoring the targeting efficiency of your CRISPR design to the amplicons, and you can basically replace the gel electrophoresis with our system. and our CRISPR gen, which basically completely eliminate the need for amplicon generation and can provide you with similar information, but without using amplicon. You're basically using the power of CRISPR in searching the whole genome to see the impact of your modification in your targeting effect. So in this video, you're seeing our first product, which is a liquid handler system. And we have developed software where we can choose your protocol. You can input your protocol, and the system basically you put your material. The system has 8, and it'll be going to 24 within the next 1 or 2 months. Chips, and then you can put different type of Cas, different type of guides, and really compare to see how changing those guide RNAs are impacting your complex formations. And we have developed, machine learning algorithms which can analyze the data and basically see what are the parameters that are impacting your design and the stability of your complex the most. So this way, you're basically removing the user variability. Everything is done by a robot and a liquid handler, and you can monitor the data, and the software will do the analysis for you. So as you see, our core technology is rapid multi-analyte. It can be rapidly reconfigured, and you can use it for many different applications, and form factors can be different for your application. And we're working with a lot of partners, from the drug discovery to AgTech to food quality and safety to law enforcement, to develop different type of product with them powered by Cardea, where we partner with them to develop those products. So make sure to visit our booth. My time is over, so I want to be respectful to my other colleagues' time. But CRISPR QC is one of the first partners that is utilizing our platform to do quality control for CRISPR. That's in terms of like how the editing work, the editing efficiency, the complex stability, and other type of quality control application. The next generation of devices that we will be generating in the next few years will be not only monitoring this type of function, but also we'll be monitoring, we'll be diving deep into looking at real-time enzyme functionality in terms of if we can monitor the enzyme in action in real time, and how does the conformation of the enzyme changes as you expose it to different type of targets. So with that, I want to acknowledge Cardea Bio, Berkeley, and Keck Graduate Institute for supporting this work. And please visit our booth. I think Amanda, she is in the chat. She will be sending you a link that you can visit and look at some of our products. And there will be a chance to win an Amazon Echo or a Google Nest. I apologize for, I think I won't have time to get to the question, but I'll get to the chat and I'll answer your question. And my colleagues will answer your question also in the chat. Thank you very much. So with that, and I promise that I'm going to get to the question and answer and get back to you in the chat. But I want to introduce my colleagues, Dr. Stanley Qi, who is a CRISPR expert and an assistant professor in Department of Bioengineering in Stanford. And he will be presenting CRISPR as an antiviral strategy against COVID-19 and RNA viruses. Thank you so much, Dr. Qi.
Thanks, Kiana, for the introduction. And first, I'd really like to thank the organizers in Synthego for hosting such a wonderful event. It's a perfect time to have one day dedicated to CRISPR innovations and also pointing to the future. I feel re-energized by looking at so many great talks. I'm Stanley Qi, and I like to talk about our recent work on developing the CRISPR as a broad-spectrum antiviral to combat potentially SARS-CoV-2 and other respiratory RNA viruses. We call the technology that we've been working on, PACMan, stand for prophylactic antiviral CRISPR in human cells. And this is a cartoon made by an undergrad in the lab to capture the general idea. It's kind of like a game to chase down the virus and kill them. My talk consists of a few parts. One is a brief introduction to the background. Then I will talk about 2 parts. One is targeting influenza A virus, and second is coronavirus, including SARS-CoV-2, and another milder coronavirus called 229E. And finally, I will point out the challenges and also the future solutions. So we've been thinking about the use of CRISPR as a potential tool to fight RNA virus. And for this reason, we choose to use the type 6 CRISPR Cas13 family, which is a specific RNA-guided RNA cleaving CRISPR protein. From the pioneer work from Feng Zhang's lab and also Patrick Sue's lab in the past couple of years, this basic research revealed this class of CRISPR as a very interesting, very efficient RNA-guided RNA targeting molecule. They can target specifically the RNA molecule in the primary manner, but also they may have some other issues like how collateral effects to target RNA, like bystander RNA. So what's really intriguing into this CRISPR-Cas13D, which is the system we used in our research, is due to its high efficiency and high specificity, as shown this data from Sylvana Kornerman's paper 2 years ago. You can see that when they use this system to target endogenous human genes, it shows really nice inhibition, more than 90% on most genes, and also very high specificity, only the target gene being significantly affected, but not other non-target genes, suggesting even though biochemically this system may have some bystander RNA cleavage activity, but in human cells, this effect has not been significantly observed. Given this system, our hypothesis is maybe we can use this system to specifically target and degrade the viral RNAs, thus providing enough time for the human body to develop immunity against virus. I find this idea interesting because CRISPR has been identified as an immune system for bacteria, has a very long history in evolutionary history. And if we can use that for human immunity, that would be a really interesting repurposing process. The question is whether it can be done or not. So 2 years ago, supported by DARPA, we started to work on using Cas13 as a potential preventive strategy to fight human influenza A virus. As you know, influenza causing flu is a really broad category of virus causing seasonal flus and mutates really quickly. And its biology is somewhat different from coronavirus. For example, it contains 8 fragments of genome, all of them RNA fragments, and it rapidly evolves and replicates in the human body. Since it has so many fragments, we started with a screening process to define different guide RNAs targeting different regions of these different fragments and trying to look into which fragment, when we target, would be most effective in inhibiting the IAV replication in human cells. It's interesting to observe that one fragment, segment 6, encoding the NA gene, and is most effective in inhibiting virus replication. And later, we verify this screening process by challenging the human long epithelial cells with both CRISPR, because we want to get a prevention here, and then later, after 24 hours, challenge them with the live influenza A virus. And after another 24 hours, we perform measurement to see viral inhibition. It's interesting that we can verify the original screening data. We observed at 2 pretty high multiplicity of infection, MOI, 2.5 or 5, which is really high, higher than actually what may happen to the human body in natural case. And we still observed pretty strong inhibition, as you see the data here. This gives us some confidence that CRISPR-Cas13, despite it being discovered as a bacterial system, it may also work effectively to fight human virus in human cells. And when we look at earlier this year, when SARS-CoV-2 COVID-19 started, we look at this biology, and we realized although the detailed genome organization of SARS-CoV-2 is different from IAV, but we may be able to reuse these tools for fighting the virus. And it has shared similar biology that the viral genome will be replicated from a positive strand into a negative strand, and using the negative strand as a template to make more copies of positive strand to package more variants. And in the meantime, and they will make a lot of viral mRNAs to make a tons of viral proteins to facilitate this process. Essentially, these proteins and RNAs take over the whole human cells. And we thought, okay, let's target somewhere in this viral gene RNA And then we can shut down both the viral genome replication and also its viral protein production. For this purpose, we choose to target the positive genome RNA in SARS-CoV-2. And as you can see from this diagram, if we target the positive genome RNA, we may be able to both shut down the replication genome because it's decayed, and second is we may prevent the mRNA from being translated into the viral protein. So we may get dual effect for inhibition. Since the coronavirus has a pretty large genome, actually it's the largest, 30 kb, and there are a lot of potential binding sites for a small CRISPR, which usually binds 22 base pairs using the Cas13. And where should we target? There are so many options here. And for this purpose, we choose the conservative regions on the viral RNA, and we looked at, we find two primary regions that may be both biologically important and also highly conserved among all the coronaviruses. One is RDRP, RNA-dependent RNA polymerase. Another is a nucleocapsid protein. And RDRP is the replication machinery. And N protein capsulates the viral RNA, which is both are essential for the whole viral replication. And we start designing guide RNAs targeting both regions and screening guide RNA which can be most effective. To facilitate the screening process, we started by constructing synthetic SARS-CoV-2 fragments by cloning several kb fragments from the SARS-CoV-2 onto the human lentivirus, which is also exists in the RNA format in human cells temporarily before it's integrated into the genome. And also we cloned pigment system onto two separate vectors. One encodes Cas13, another encodes a category of different CRISPR RNAs. We challenged these two systems to human long epithelial cells, first with different CRISPR RNAs with Cas13D, and after the transfection or transduction process, we challenged it either using the synthetic SARS-CoV plasmid or synthetic lentivirus, encoding the SARS-CoV-2 fragments and measure gene expression and also RNA abundance. For example, just one example here, because there are so many binding sites, we started testing pools of guide RNA in pools of 4 guide RNA combined together. As you can see, when we measure both gene expression from the lentivirus and also measure the RNA abundance from the same sample, we can see some groups performed much better than other groups. Some groups have no effect, some groups have a decent amount of inhibition. Then we took the most effective group, for example, group 6, and decompose them into individual guide RNAs. To our pleasure, we find in doing that, we can rapidly define CRISPR RNAs that has a much higher efficiency in inhibiting lentiviral form of the CRISPR RNA, the SARS-CoV-2 fragments, almost up to 98%. And in the meantime, I help bioinformaticians start asking questions. Why we are using CRISPR, right? So that's an interesting question because it's a genetic system. It can be used to target conserved regions, and it may provide pan-coronavirus inhibition if we carefully design our guides. So this diagram captures the basic idea. We want to do a map using minimum amount of blocks to cover this whole space of the coronavirus. And as you know, there are more than 3,000 coronavirus has been sequenced so far. Only 7 infecting human, including SARS-CoV-2. And all other 3,000 either in the bats or in other mammals. And someday they could evolve and start to infect human, or new strains can be recombined to become infectious in our society. So using the bioinformatic tool, we ask questions, what is the minimum number of CRISPR RNAs we can design to provide the broadest coverage of these 3,000 coronavirus? And here is a bioinformatic analysis. As you can see, to target 50%, we only need 2. And if we want to cover more than 90%, we need 6 CRISPR RNAs mixed in the pool. And these 6 RNAs, the sequence shown here, covers all the human coronavirus. And we continue to look into other human RNA virus, just in a theoretical manner. And by looking at different families of the RNA virus reported by different databases. And here we only look at the human infectious strains. Coronavirus, there are only 7, and we can theoretically design Y to target all 7 human coronavirus. And influenza is more diverse, and we know more than 90,000 strains so far, and it requires a higher number, like 6. But still, the point is, using a small number of CRISPR RNAs, we can target broader strains within a family. And we even combine all these families together and find a group of 14 guide RNAs can target all WHO-defined human infecting RNA viruses, as shown in this diagram. Of course, because many R viruses are infecting human blood or human other organs, liver, it may not be amenable for delivery method at the moment. But we are glad to see theoretical analysis shows CRISPR, because it's a genetic system, it has features that are not possessed by other antiviral, like protein-based, antibody-based. And also, it's interesting to look into biology, why some requires much more than others. It could be due to the higher divergence of the virus, or because a lot of members strains in that family. So now there are some remaining questions. Can Cas13 inhibit other coronavirus? Means is kinetics fast enough? Cas13 will kill virus, but virus will replicate, right? Who is faster, and can that be effective? Second is, how can we deliver? And what is third, what is off targets? We start looking into these questions. And for this purpose, we're looking at one virus by one virus. And several of them are BSL-3. And at Stanford, we just only had our BSL-3 recently. But in the meantime, we've been testing BSL-2 virus. In particular, look into the 229E strain, which is a BSL-2 virus. And for example, using the live BSL-2 229E virus, we try to define different guide RNAs. Similarly, we define 40 CRISPR RNAs targeting 229E, but this time not using synthetic assays. We start using the real virus by infecting a human long cell line called MRC5. And when we look at that, we find the best CRISPR RNAs can inhibit almost 97% inhibition of the 229E after 2 days post-infection. And after 3 days post-infection, when we take the microscope images, as you can see with the CRISPR RNA pacman, many cells are very healthy. And you can see nice gene expression and morphology, but without many cells start to die or have really weird growth effect. So this actually suggests within a short period of time, the pacman can rescue both viral inhibition and also cell growth. Now we start collaborating with biochemists to look into several methods for delivery of the pacman into human cells. And with ideal final goal to deliver in vivo as a therapeutic modality. One modality we look into is called the lipidoid, in collaboration with Michael Connolly from Berkeley and Annelies Barron from Stanford. In the past 2 decades, they've been looking at this really interesting chemical structure called the lipidoid, which is an amphiphilic molecule that can penetrate the human cell membrane and deliver payloads. And it works really well for RNA delivery. And to our pleasure, when we test Cas13D, which is a large mRNA, 3 kb, and plus mixed CRISPR RNAs, we saw a decent amount of delivery, almost like 60 to 70% delivery, and also the nice inhibition. This is all done in vitro, and we look forward to test in vivo. And I should also mention that we've been maintaining a very nice collaboration with Synthego, Kevin Holden, to use Synthego manufactured CRISPR RNAs. I really should say we are thankful for this CRISPR RNA because its efficiency is much higher than in vitro transcribed CRISPR RNAs made by ourselves. And in the meantime, we also collaborate with other industry partners to test not using transfection reagents, but really using nebulizers. A picture shown here. To our pleasure, we find nebulizer because it can generate mist, almost like aerosols, and which can allow even better penetration. An example is shown here. Negative control shows no delivery almost. If we put like just a transfection, this is reporter, so it's higher, like 90%. But if we use nebulizer, it's almost 99.8%. We are very excited about this possibility of using several already demonstrated industrial products to further facilitate our development of pacman. So this is one of my last slides that I would like to talk about. So when we think about CRISPR, why it's intriguing? Because it can be used as a broad spectrum antiviral. I keep asking myself why we are not so afraid of bacteria. Of course, I'm afraid of bacterial infection, but we have a lot of broad spectrum antibiotics to kill them, and they are off shelf. And they also escape a lot of evolution escape. We don't have similar things for antiviral. We hope CRISPR can be used as such an antiviral. So to cap my talk, I think pacman is emerging as a new system that's worth testing for therapeutics or prophylaxis for RNA virus. Its efficiency is pretty nice, and also we think there are a lot of could be there for broad spectrum antiviral. With that, I'd like to thank my collaborators. I cannot name every one of them because they are all contribute significantly to the project and also the funding agencies. Thank you. I'll be happy to answer questions also in the chat channel. And also sorry for being a little bit longer on the presentation side.
Thank you so much, Stanley. Thank you very much. So our next speaker, it's doctors Gootenberg and Dr. Abudayyeh from McGovern Institute. Dr. Abudayyeh and Dr. Gootenberg are both co-founders of Sherlock, one of the first companies that actually brought CRISPR into diagnostic realm, and they started, everything started from them, I think, and they're the paper that they published. Good to see you guys again, and
hey, nice to see you.
And I'll give it to you guys so you can start presenting, and we don't go short in time again. Thank you so much.
Okay, I will try to bring this up. Cool. So I guess, yeah, Omar, will sort of give the first half of the presentation, which will focus on a lot of our diagnostic work and what we've been doing for COVID-19. And then I'll pass it on to Jonathan and talk a little bit about our RNA editing and mammalian cell applications of these tools. So kind of see both sides of the CRISPR tool set. So just to dive in, a lot of our work and what our lab sort of does now is really look for problems in either cell engineering or genome editing, and as well as now diagnostics, and tries to really solve them by mining for new types of proteins and enzymes within the bacterial world. And we do this a lot for CRISPR, but now we've also been trying to look beyond, because there really is a lot of rich diversity in bacteria and other organisms. And I think within the CRISPR world, when we started many years ago looking at CRISPR diversity, Cas9 was really the only single protein system that really existed. And of course, you can see it here, but there really was a lot of other diversity known at the time that a lot of people have been studying for many years. And we wondered whether there could be more diversity for these simpler looking systems. So of course, to sort of go down that route, we had to build signatures of what a CRISPR system could be in order to find new ones and sort of all the new sequencing data that was coming online and being deposited. And so for our initial discovery, we focused in on the highly conserved sort of adaptation proteins like Cas1. Of course, you can also use the CRISPR array as another sort of signature of where there might be CRISPR proteins nearby. And we found a number of new systems like CPF1, which is now sort of the first Cas12 family member, Cas12a, and now there's been many, many Cas12s found. I lose track of, I think, what subletter we're on. It might be J. I think it's Cas5s. So, but it's kind of cool, like how much diversity there is in just these RUBC containing enzymes. And then, of course, the most interesting one that we have spent a lot of time with is CQC2, which was sort of renamed to Cas13. And what's interesting is it really didn't have any sort of DNA targeting domains. Instead, it had this sort of HepN domain, which turns out it's an RNase domain. So, we spent a lot of time characterizing the mechanism of these proteins, and it turns out, it's a programmable RNase. It's not precise like Cas9. In fact, when it binds its target, it generates a lot of cleavage sort of sites and can actually degrade RNA. And turns out it'll not just cleave the target you're bound to, but it'll cleave a lot of other targets nearby, which at the time, since we were so focused on RNA therapeutics, we were like, oh, that's too bad. But it turned out, us and others, I think, realized that this could be really useful for diagnostics. And so just to, as I'm sure many people are aware now, you can use this for diagnostics by, designing a guide RNA, let's say, against a blue molecule shown here that you want to detect. And the idea is if you have an RNA reporter in solution that has a fluorophore quencher, you can basically get cleavage of that when the Cas13 binds its target, and it'll release fluorescence. And this is, pretty sensitive by itself. I mean, it takes, millions of, you still need like millions or tens of millions of molecules per microliter to get detection of the system by itself. I think, was it Jennifer Doudna and Melanie Ott, recently published that if you design many guides along a target, you can actually use this system to improve the sensitivity a bit, but it still doesn't get down quite to single molecule. And to do that, we've had to combine other technologies like preamplification technologies like RPA or LAMP into the reaction or upstream of the reaction to get the sensitivity levels that are clinically relevant. Now, we as we've sort of been pulled into the diagnostics world through a lot of these unique sort of biochemical mechanisms, there really is a lot of need there. So both within the infectious disease, as I'm sure we all realize, since we're on a sort of Zoom call right now, there's a huge need for like rapid point of care infectious disease detection, but also just in other areas like genotyping, doing cancer detection, even outside of healthcare for agricultural, tracing traits in like plants and whatnot. So I think that's a good segue into kind of our newer work, which started at the beginning of this year, which in January, I think we started hearing about all these sort of cases coming out of Asia of a new weird pneumonia-like illness, and I think us as well as Feng, who we've been collaborating with, we're getting a lot of messages about what can you guys do with CRISPR detection and Sherlock. I think after a couple of weeks of just basically daily texting with Feng and ourselves about like what should we do, we dove in, and I think this is must have only been 2 weeks, but we basically posted this like white paper protocol about how to use our typical CRISPR detection, which is a two-step reaction with preamplification and CRISPR detection, and then a lateral flow strip. And we actually posted it and then shipped like a ton of kits all over the world for people to try it and play around with it. And it worked pretty well. I mean, we could on patient samples actually detect I think close to single molecule. It must have been around 10 or so molecules. And a lot of people, we've collected a lot of nice clinical data with this approach. Now, March hit, and I think we realized that to really, basically the whole world was going to need testing capacity. And, this approach, was working well. There was actually a commercial test. I think this was maybe from Sherlock Biosciences, but basically they were able to get their two-step chemistry authorized, which is the first CRISPR healthcare product kind of in the market, but for central labs and CLIA testing, which is great. But as we now know, like the FDA today is not even approving any more central lab tests because we have way too many of those, and they're bottlenecked by the processing and the turnaround times. And what we really need is point of care at home tests that are scalable. They're scalable because anyone can run them, so you don't have to wait for a central facility to process your test. And they should be cheap enough such that you can run them every day or whenever you are worried about your health status or need to go to work or just go shopping. So our vision to basically take the next step, which is how do you combine everything into a single reaction in a very simplified format. And we actually switched from RPA to LAMP because we find LAMP to be much simpler and easier to scale as a chemistry. But then that posed a problem because it's 60 degrees, it's hotter, and there weren't any collateral-based enzymes we have worked with that work at hotter temperatures. So we actually found this new enzyme that was basically from bacteria that comes in rotten fruit juice. So if you leave your fruit juice out in the kitchen, there's this bacteria that'll have this new Cas12b in it that can survive acidic environments, and those properties also make it really thermophilic, and so it actually works at 60 degrees. And we combine that in the same reaction to basically cleave a DNA reporter that gives detection. In this case, it says FAM biotin, but it's actually FAM quencher if you want to do fluorescence. So to pull all of that together, which was actually we published this a month ago in NEJM, but the idea is it was a very simplified format. And not only did we have that single sort of one-step master mix, but we combined it with a simple extraction step upstream so that you didn't have to do RNA purification, which is another bottleneck for the central lab-based tests. And so the idea is you basically can swab yourself, you put in an extraction buffer, the beads bind the RNA that's released, and then you resuspend the beads in our master mix. And then you heat it for 30 minutes or so, and you can do lateral flow or fluorescent detection. And just to show why CRISPR detection is useful, LAMP by itself will have a lot of non-specific amplification, but the CRISPR detection really makes it quite specific and takes away any non-specific amplification. So it's a common question we get about the use of the CRISPR probe. And then, of course, on a set of more than 200 patient samples, we get very nice detection, and the time to threshold actually tracks the qPCR detected viral load with longer times for lower viral loads. And overall sensitivity in this data set is about 93%. Just to make it clear about how we think that's actually really good for point of care is we have a lot of 38, 39, and 40 viral load samples. in our sample set, which a lot of people who are doing evaluation of their tests don't even have stuff that low. Usually it's like 35, 36. So if you actually compare ourselves to other point of care tests on the market, the data sets we found for them actually have 36 or below, and we're actually 100% when you're 37 or below. So we're pretty happy with the sensitivity, especially for an assay that's simple enough to be point of care. So with that, I think we're excited to figure out how to move that chemistry to the next step. We're thinking about ways that it can be put on a simple system or maybe even device one day to be released. But I'll turn it over now to Jonathan for the mammalian.
Cool, thanks. Yeah. So I mean, it's a hard act to follow, but basically, one of the great things about these tools, as you actually probably saw from Stanley's presentation, was that, there's tons of RNA, both outside and inside of the cell. So, can we use Cas13 also as a tool inside of the cell to perturb the various types of RNA? Of course, the mRNAs, the microRNAs invading, viral RNAs. And so you can actually apply this to many different things. So we can do a lot of different types of RNA therapeutics. Of course, we can do RNA knockdown, but we can also do base editing. So we can actually recruit different enzymes to RNA to modify the information and create essentially transient modifications that can have a lot of interesting effects. So I think that you probably actually already got a great rundown of RNA knockdown. So given the time, we're just going to talk about base editing today. But there's many applications that you may not want to have permanent modification. So when you think about RNA editing versus DNA editing, there's many things you can do, like modulate protein function and actually change the activity of an enzyme, or correct a Mendelian disease without potentially permanent off targets. That could be useful to have an RNA editing modality. So how does RNA editing work? Well, it takes place naturally in your body through a couple different mechanisms, but a very common and basically vital one is the deamination of adenosine to inosine. So if we have a deaminase, it can actually just convert this amine group to an inosine, which is read as a guanine. So all we have to do is take that ADAR, which is adenosine deaminase acting on RNA, and we take the catalytic domain and fuse that with Cas13b. This is a catalytically inactivated one. And then we can actually take a guide RNA, and it's a special guide RNA with a misflip right here. So we can specify that target A by having a mismatched cytidine across from it. And then since this mimics the natural substrate for ADAR, we can actually convert that adenosine to an inosine, or basically have an A to G conversion in a very programmable fashion. And so we call this technology repair, or RNA editing for a programmable A to I replacement. So what does this actually look like when we do it? So this is a kind of schematic of a target. So this is a just reporter transcript, and we have introduced a premature termination site, so a stop codon that can be corrected to a tryptophan by modification of this A to G. So we can modify a couple different parameters here: how long the guide is, as well as how far away this C that we program is from the DR. And we can actually tile across and try a couple different basically iterations of this design. And we can find that at certain sweet spot mismatch distances, we can actually get fairly high levels of editing. So this is essentially 25 to 30% editing at this site in the transcriptome. Now, one thing you might notice is that there are some off-target effects here. So recruiting the ADAR will have an endogenous activity on just adenosines in general. So we saw that, and we wondered, what if we profile this transcriptome wide? So what we did is we profiled the transcriptome wide, and we found actually, at a certain coverage, thousands and thousands of off-targets. So here's that on-target editing here. We actually got pretty good 80% editing, but we can see we have all these off targets. So what we did is we actually took a rational approach where we mutagenized a couple different residues to find an optimal construct, and which we call repair V2. And you can see actually that this repair V2 dropped the number of off targets by 1,000-fold, essentially. It did have a little bit of on-target activity that changed, but in general, we find this to be a great compromise. And just to zoom in on that site, this cryptidinoluciferase mRNA that we were editing before, this is our on-target, and we've got these off-targets all peppered around. And then when we actually do this repair V2, we get full product purity. So it's a great way to actually get this to edit efficiently and specifically. So with that kind of hill conquered, we started thinking, well, how do we expand the RNA editing space? And if you think about RNA editing as the capacity to transform one amino acid to another, there's a lot of interesting things you can do, both with repairing mutations, but also if you look at things that are like phosphorylated, maybe you can take a serine to a leucine to block it, or a threonine to an isoleucine. Now, one caveat is that these are edits that are not made by an A to I editor, but a C to U editor. So this led us to think, how do we expand this targeting range of RNA editing? And the ADARs are actually, not the only deaminase family. They're actually quite similar, you can see, to the cytidine deaminases. And you can actually do a structural alignment of the human ADAR domain with an E. coli cytidine deaminase. You can find, hey, it maps pretty well, but there are some issues with certain blocking residues when you try to dock that in the pocket. So we thought, well, how do we evolve a C to U editor? And we went through a whole campaign for this, where we basically, well, first we had to come up with a little cute name. We call this Rescue. But the concept is that we can actually do iterative rounds of directed evolution in both yeast and mammalian cells. And what's that look like? So we use a similar reporter. It's on a different transcript. It's Gaussian luciferase, but we're editing this little guy here. And you can see that when we start out, we basically have no detectable editing at all. And by doing 16 iterative rounds of directed evolution, we can get this to basically 70% editing at this site. Other sites, we can go from 0 to 4%, and it is motif dependent, but it's actually quite robust across a large range of endogenous transcripts. So here we're editing KRAS, PPIB, SMARCA4, a set of, a couple different targets here, but also beta-catenin, which it was an interesting one. So you can see that we're getting basically, between 10 and 35% editing across, but say in generally about 20% editing. But beta-catenin is an interesting target because it's a very interesting part of the Wnt signaling pathway. And it's been a very interesting drug target because it's involved in things like regeneration and obviously cancer. So normally beta-catenin in the absence of Wnt signaling is phosphorylated and degraded, but if you remove that phosphorylation by Wnt signaling, or maybe you mutate that phosphorylated residue, then you can stabilize that beta-catenin. It can translocate to the nucleus, and it can activate Wnt responsive genes for things like regeneration. So what we did is we took the N-terminus of beta-catenin, and we actually targeted rescue against many of these residues. And the thought is that, if we can stabilize that beta-catenin, it'll allow it to build up and activate these, and it actually leads to increased growth, which is a really cool phenotype because you can imagine like regenerating someone's liver. So what you can do is we target rescue against these different residues, especially T41I, and we can see that actually translates to a beta-catenin activation by a transcriptional reporter. Then we can actually look at a model where this is a plug that we remove of cells, and then they grow in. And you can see here that when we target T41I, they actually invade much more, significantly more than the non-targeting case. And you can actually quantify this by light microscopy. So it's a really interesting case where we can actually use this therapy potentially to do things like organ regeneration. And this is a place you wouldn't want a DNA therapy, of course, because you wouldn't want these cells to proliferate forever. But a cool thing is, as this technology, as these cells are proliferating, depending on your delivery mechanism, they'll actually dilute out. So it's a natural kind of kill switch that comes along with the technology. So we can use this for a whole slew of different things, like correcting Mendelian diseases, doing multiplexing, which we show on the ApoE target, and then modulating catalytic activity or post-translational modifications in a way that couples, I'd say, transience of a small molecule with the precision that you've seen throughout this conference of genome editing and other editing modalities. And going even higher level, RNA targeting and RNA binding proteins have a huge range of applications where we can, modify translation and exons, modify localization, even as many people have done, do epitranscriptomic modifications. So we think of this as, compared to the DNA editing toolbox, the RNA editing toolbox is, I'd say, not there yet, but it's definitely growing by leaps and bounds. And just to bring it back to the tree of life, we're still very interested in, how we can mine this for great resources for biotechnology. So it's something that we're really inspired to do in our new group. So for the people who made this work possible, we'd like to thank our advisor, Feng Zhang, who helped us a lot with the early Sherlock work, and we collaborated with on the Stop COVID, as well as we did the RNA editing work in his lab. And we'd also like to make a little plug for our new lab, which is at the McGovern Institute. We're actively looking for postdocs and excited people to join. We feel free to reach out. And we have our lab website at abudayyeh.mit.edu. But yeah, with that, I think we have time for maybe 1 or 2 questions. And thank you for listening.
Thank you so much, guys. Thank you very much. And I think I'm going to ask one question from the audience, and then we have to lead to the next session. So Rebecca asked, she loved the stop COVID test. The sensitivity is impressive in a scientific, technical sense, but the real world application, how should low-level positive guides decisions? Or basically, I think she's asking about the PCR need and the amplification need. How would you see your technology to be amicable to those resource poor settings?
Yeah, well, I think our technology fits really well in research poor settings because, first of all, it's very easy to run. We actually had a figure in the paper where we used a sous vide like water bath like that you could run in the kitchen to basically heat the whole reaction. And you don't really need like fancy pipettes. You can actually just literally use a dropper to transfer, the extraction buffer into the solution. So, but we are working on like a very simplified sort of system that can just run the assay with some basic optics as kind of a next step. And we're hoping to actually have that released in the near future with hopefully an EUA as well. So I would stay tuned for some of that work. So
perfect. And I'm going to ask one more question because we started a little bit late. I'm going to ask Mino to give us a few more minutes. But how do you ensure that self-testing is compliant? Like if there is a little bit of variation between how the users are using the system, do you think that could cause problems, especially if you're doing some sort of a diagnostic between 2 or 3 different types of viral infections?
Yeah, I think that self-testing is like a huge thing, and we think about usability and adherence. How do you make a workflow that's robust to things? If you have testing of different inhibitors, that can be at different places in the nostril, for example, even. And how do you make it so you get things come off? So we're doing lots of usability testing to make sure that it's a robust platform. But that's a very real-world concern, and we're working hard to address that.
Thank you so much. There are many questions for you guys. I think if you can go and answer those questions, that would be great. Thank you all very much. I encourage everyone to attend the next session, which is about the future of CRISPR and engineering and educational challenges. It's an exciting session because you're going to talk about with producers and scientists from the CRISPR documentary film, which is now available on Netflix, the Human Nature. They're going to talk about and discuss and share insight about what the future of CRISPR holds for us. So I really encourage you to definitely attend the next session. Also attend our booth so you can see some of our robotics products for CRISPR-based transistors. And with that, I'd like to end the section and hope to see you guys again soon in future conferences.
Great. Thank you so much.
Great.
Thank you all.
Thanks so much.